Can we start now? So how many students are having this NET exam today? So almost all of you, or 50% of the class, or what?
Sir, actually, the students who are taking it are not present.
So could it be, oh, half of the students? All right. Okay, so maybe initially we can talk about if you have any issues that we need to deal with. So that means your questions. And obviously, because this is the last day of the week for class for BIO404. So whatever we cover by today, so I'm going to upload on the Moodle page. Now tell me, so is the Moodle page working now? So are you able to access Moodle?
Yes, sir.
Well, looks like last week there was some issue. I don't know what was it. There was email from computer center that somehow this is not available. Anyways, questions? If no questions, so let's get started. Okay, so because many of you are not present today, so from your batchmate, so we may not cover quite extensively. Okay, so maybe some part of it. So we'll try to complete the topic we started, and then we are not going to actually discuss it quite elaborately.
Recording in progress.
Okay, so yeah, so we were talking about monoclonal antibodies, and the procedure involves injection of an antigen into a mouse. Mouse is going to generate an immune response. So essentially that means generation of antibody producing cells. So to assess whether the animals which were injected with this antigen, okay, mouse or mice, whether they have induced antibody response, so you collect some serum sample. If antibody response is observed, so you sacrifice these animals, collect spleen and other lymphoid organs, make single cell suspension from the splenic tissues, right? You simultaneously you culture tumor cells that we call as myeloma cells. You fuse myeloma cells with splenocytes, right? Obviously, for this fusion event to occur, so you have to add some chemical. In this case, this can be polyethylene glycol. There are some proteins derived from some of the viruses which can be used. You can also then you are going to actually observe cell-to-cell fusion, generation of these hybridomas. Then you have to select these hybridomas. screen for their ability to produce antibodies. This actually process, as we discussed, require quite extensive analysis. But eventually, so if you are able to establish that this hybridoma is able to produce antibodies which are reactive to your antigen, you actually can generate monoclonal antibodies, right? And we also talked about this slide where you have, for example, these plasma cells coming from immunized animals. They were fused with myeloma cells. There are two characteristics of these myeloma cells. One is they should be deficient in antibody production. They should also be deficient in this enzyme, HGPRT, right? Why they have to be deficient in this is because this will help in this selection process using this het medium that we are using for selecting only the hybridomas, right? Or the fused cells. Okay, so you can actually generate different types of fusion, or you might get unfused cells. So for example, if you have only primary cells which are not fused with cancerous cells, so they are going to die because of senescence. So if you have homologous fusion between primary cells, again, they are going to die because of senescence. Senescence is because after a few rounds of division, the cells do not sustain because these cells are not cancerous cells, right? And you can also have the unfused cancer cells, which if you supplement this medium with het, this is het, okay? So we'll talk about the composition of this medium. So this will basically remove unfused or fused cancer cells, okay? Because they are deficient in this HGPRT, right? So eventually you would be able to select only those cells or the fused cells from primary and the cancerous cells. They can sustain in this medium by utilizing the salvage pathway for nucleotide synthesis. And you know, nucleotides are required for replication of DNA and several other processes, right? So they are not going to have this senescence because the immortalization is provided by the cancerous cell. So you are able to select only the fused heterokaryon. So that means the cells which consist of primary cell as well as the cancerous cells. The fused product of these two things you would be able to select out.
Sir, what is the full form of hat medium?
Yeah, we'll talk about it. Okay, so in 2-3 slides, so we are going to talk about all those things. Now, so you know that when you actually are fusing these two cells, right? One of them actually is a primary cell, one of them is a cancerous cell. We did talk about that you are starting with a total splenocyte, so you can actually generate hybridoma for T cells as well. And if you generate hybridoma for T cells, the purpose is that you can actually produce a lot of cytokines. For B cells, the purpose is that you actually can produce antibodies which are of defined specificity. Okay, so if you make T cell hybridoma, so you can make abundant of cytokines, okay, which are actually produced by these T cells. So you are fusing one primary cell and one cancer cell with this agent that we call as polyethylene glycol. Basically, this is a very viscous material. So this brings these two cells together, and through membranes, there is a fusion of the membrane of these two cells, right? And whatever the nuclear content you have, which is coming from either primary cell or cancer cell, they are going to actually fuse then. Okay. At this stage, basically, you call it as heterokaryon, where the cell membrane is fused, but you have these two nuclei, right? Eventually, you are going to get the nuclear fusion as well. Now, question here is, because you are already starting with the diploid cell, right? Your B cells are diploid, your cancer cells are diploid, right? The myeloma cell that you're using for making hybridoma. So these are actually diploid cells, right? So you cannot really work with 4x number of chromosomes. Okay, so randomly, so one set of chromosome has to be actually lost from these cells if they have to survive. And this actually is a random event. And that is why the success rate of generating these monoclonal antibodies through hybridoma technology actually is less, because there are so many variables in between, right? For example, a tetraploid cell is not going to survive, right? This is not normal for example, for these diploid cells to have two sets of chromosomes exactly in the same cell. You know about the mitosis and meiosis, right? So first the chromosome have to be divided, they have to make 1x, and then basically in the gametes, then you make as 2x or diploid, right? And here, so you are basically starting with diploid cells, and when the nuclei of these cells are fused, so you are going to generate tetraploid. And this cell cannot really live for sufficient duration. So if this cell has to live, so it actually has to lose one set of chromosome, and that occurs in a random fashion, right? But let's say now if you are able to generate a hybridoma cell, which again has only diploid architecture, right? So the genetic material actually is diploid because you already have lost one set of chromosomes, right? This cell you are going to select and characterize for its ability to, one thing is survive, second is to produce those antibodies, and then you should be able to characterize what antibodies are being produced by these cells using different immunological assays, starting from ELISA to Western blot to whatever the analysis you want to do. And once you establish that a fused product of your B cells and cancer cells now is able to produce this antibody of a particular specificity, so now you have a hybridoma, which you can grow almost indefinitely. And this will keep on producing these antibodies. And you literally have actually a source of antibody producing cells. Whenever you want to grow this cell, you can grow, and these cells will start producing antibodies, and then almost inexhaustible supply of antibodies can be produced from these cells, right? And once you have a monoclonal antibody, so you can use it for different purposes, right? So this slide actually shows that you can generate B cell hybridoma. You can also generate T cell hybridoma. The purpose of B cell hybridoma is to produce these antibodies of a defined specificity. Purpose of generating T cell hybridoma is to produce cytokines such as IL-2, right? Yeah, these are some of these things that you need to know by taking example of one of the myeloma cells. Okay, so we said that these myeloma cells, they have to be deficient in this enzyme HGPRT. The full form is hypoxanthine guanine phosphoribosyl transferase, right? Or alternatively, you can have a cell line, a myeloma cell line, which is deficient of thymidine kinase. So it will come in next slide why actually this is required. And one of the major reason is this cell line you can select using this head medium. Okay. Now cell line cannot survive in selection medium. Okay, because of deficiency of these enzymes. These are actually very critical for synthesis of these ATP, GTP, and all those nucleotides, right? The medium carries this compound. This is basically antifolate. Okay, so this compound actually is called aminopterin. Okay, so just remember that this is one of the component of this het medium. Okay, aminopterin, and it inhibits de novo pathway. And salvage pathway is not possible in these cells because they are deficient in these two enzymes. Okay, so essentially, so this is going to get rid of all the cancerous cells, except for only the fused product. Okay, because when this cancer cell is fused with a primary cell, so this can be selected in this medium because this enzyme is coming from your primary cells, B cells or T cells, right? So in next slide, so we'll briefly talk about these two different pathways to make things more clear. The other property of this myeloma cell, this is actually one of those things, P3.653 myeloma cell. This is a very commonly used cell. Okay, this is basically a common cell used for making antibodies. Now, one more thing that we talked about was that myeloma cells cannot serve as the source of antibodies because antibody production has to come from your B cells, right? Because myeloma cells can keep on producing this antibody, but you don't know the specificity of this antibody against which antigen the antibody is being produced, right? So you have to have a B cell which is induced in animals following immunization with an antigen. So that means these animals will have enriched population of B cells which are able to produce antibodies against the injected antigen. Okay, now this, you cannot really contaminate the production of antibody by B cells and myeloma cells. That's why myeloma cell has to be deficient in its ability to produce immunoglobulins. Okay, aminopterin is basically a folic acid antagonist which blocks de novo pathway. And once de novo pathway is blocked, so this actually helps in the selection of your hybridoma cells through cell-based pathway. Cancer cell alone will not be selected because they are actually deficient in these two enzymes which can utilize HGPRT or TK, thymidine kinase. Okay, so to summarize it, these myeloma cells, they die in the presence of aminopterin. They cannot utilize cell-based pathway because they are deficient in this enzyme, right? However, if they are fused with primary cells, so they can actually be selected using this het medium by utilizing cell-based pathway. So these are two pathways for synthesis of nucleotides. Okay, and we said that these nucleotides are required for incorporation into newly replicating DNAs, right? All right, so this is basically broadly, you can classify these two different pathways. Okay, essentially the purpose of these two pathways, de novo synthesis of purine nucleotide and cell-based pathway, is to generate those nucleotides, either purine or pyrimidines, right? So in de novo synthesis pathway, so basically the precursor is 5-phosphoribosyl 1-pyrophosphate. Okay, now this enzyme is blocked by aminopterin that you're using. The other name of this aminopterin is antifolates. Okay, now if, for example, this aminopterin is not present in this medium, so through different steps, so PRPP in short, this 5-ribosyl 1-pyrophosphate can generate inosine monophosphate, okay, which then be converted into either guanylate or adenylate, AMP or GMP. And GMP are then is converted into GTP, AMP is converted into ATP. Okay, and these are the nucleotides which actually are required for nucleic acid synthesis. Okay, now with the de novo synthesis pathway, so if you add aminopterin, so this will block this pathway. So this pathway is not going to work. So now these cells actually have to survive only with the salvage pathway. Salvage pathway is basically an accessory pathway which can generate these nucleotides from precursors such as hypoxanthine. Okay, hypoxanthine is converted into inosine, and then GMP and AMP are formed. Okay, so you can also have this guanine converted into guanylate, and adenine converted into adenylate, and similarly for pyrimidine, so you have this thymidine converted into thymidylate, TMPs, right? So now, if you think about it, you have your primary cells, you have your cancer cells, right? Cancer cells cannot really survive in presence of this antifolate you are using, the aminopterin. Okay, so that means they cannot really utilize this pathway, but they can still survive in presence of, because they can, as a compensatory mechanism, they can use salvage pathway. But remember, so you are using this cancer cell which are deficient of HGPRT, so this enzyme. Now, if this enzyme is not present, or thymidine kinase is not present, so even this pathway will not be utilized. And if this is not utilized, so that means the cancer cells actually are going to die. If you have only the cancer cells or cancer cell fusion, right? So those cells are not going to survive. And you already know that primary cells, after a couple of rounds or few rounds of division, are going to die because of immunosenescence. So the only thing survive in this selection medium is the fusion product of your cancer cell and your primary cell. Primary cells are producing antibodies, and cancer cell is actually providing this immortalization, right? Now, because this is a fused product, this is a hybrid, okay, your primary cells can still have this HGPRT, but not the cancer cell. So this fused product can survive, but the cancer cell alone cannot survive because they are deficient in HGPRT. So this pathway this is actually the biochemical pathway for selecting only the hybrid cells. Okay. Go through these different steps that are enlisted here, and you would understand how biochemically you are able to select out only the fused products, and obviously not the primary cells because they die because of senescence, and cancer cell because they are deficient in HGPRT, so neither they can utilize this pathway because you have included aminopterin, nor this pathway because they are deficient in HGPRT. Okay, this is basically the written form of whatever I just have talked about in the previous slide. So you can go through this slide. So these are two different pathways: de novo synthetic pathway and salvage pathway. And this actually is required for synthesis of these nucleotides for DNA replication to occur in the cells. And if DNA replication is not happening, so even your hybridoma cells or even primary cells or cancer cells are not going to survive. Okay, so basically you make sure that you provide a situation or a condition in which your primary cells die out because they get exhausted after whatever 3-4 rounds of application, they die out. Your cancer cells are not surviving because they are HGPRT deficient. They cannot utilize salvage pathway. The only thing which is selected is the fused product. Okay. Now, there are actually many diseases which are known to be associated with deficiency in this nucleotide metabolism. For example, if you have purine metabolism problem in your cells, so you start developing these diseases. Deposition of uric acid, clinical gout. Gout is basically deposition of these uric acid in your joints. The neuropathies, neurological diseases, and basically which actually can be even self-injurious. So basically, your brain is not working that efficiently. Okay, so these are actually very important pathways for synthesis of these nucleotides, and they are associated. If, for example, you have deficiency of one enzyme or the other in individuals, so they will have one problem or the other, right? Okay, after, well, any question on monoclonal or polyclonal antibodies before we briefly talk about a unique antibody type? Okay, if not, this is the antibody we talked about, the conventional antibody, and is generated by this DNA rearrangement. So we are going to talk about these processes of VDJ and VJ recombination to generate the variable region of heavy and light chain, which actually form the paratop, the antigen binding site, and then you have the two light chain and two heavy chains, and heavy chain and light chain also have the constant region responsible for different functions. Okay. However, there is actually a one antibody type which is naturally existing in some animal species, and they actually have revolutionized the whole field of antibody engineering. Okay, so these are called actually heavy chain only antibodies. Okay. So basically, these antibodies are dimer of only two heavy chains. They don't have light chain in the structure. Okay. And if you actually look at the structure, so even the heavy chain actually is truncated. So it really does not have this constant domain, the first constant domain in the heavy chain. Okay. So they are much shorter than your conventional antibodies. The size of conventional antibody is how much? For example, if we are talking about IgG in humans or mouse, so what is the molecular weight of an antibody molecule?
Around 150 Dalton.
Yes,
Kilodalton.
Kilodalton, 150 kilodalton, right? The size of these antibodies actually is close to 100 kilodalton. Okay, now 100 kilodalton and probably 150 kilodalton is not that much different, although you have 50 kilodalton difference. Okay, but there is another property. Okay, because you don't have actually the constant region in the domain 1. Okay, this domain actually is missing here. Now, if you don't have this domain, right? So essentially, actually in this domain, so you have those disulfide bonds, which actually make these dimers, right? Or rather, basically the which can actually make disulfide bond with the light chain. Okay, so if this domain is missing, so it really does not have anything genetically which can give rise to a product which can interact with light chain. So that is why light chain is not present. The other thing is, if this domain is missing, so the FAB portion only has the VHH. We call it as variable region. For the conventional antibody, we call it as VH for heavy chain and VL for light chain. Okay, so here, so you have the variable region of these unique kind of antibodies. We call it as VHH, variable region of the heavy chain of heavy chain antibodies. So VHH in short. Okay, so now it's very easy to genetically engineer one single domain, then the one domain of light chain, one domain of heavy chain, and then you have to basically make this whole thing from the other chain, right? So if you try to make a genetically engineered antibody from a conventional antibody, right? So this is called a single chain fragment variable. So where all the variable regions actually are in one chain, right? VH, VL, and then 2 times. So you actually have to basically use a lot of genetic linker to make it as a single chain, right? Now in this case, so you don't have that problem because light chain is not present, and this actually exists naturally. So a lot of heavy genetic engineering is already done on these antibodies in natural form. Okay, you can do a lot of genetic engineering in the conventional antibodies to make the single domain, but these products are not stable. They are not soluble. They do not resist robust biophysical and biochemical conditions. Okay, but all these good qualities actually are present with these antibodies. So that is what I said. So with the discovery of these antibodies in some animal species, starting from nurse, sharks, camelids, all the members of camelid family, so they have this kind of antibody. So they actually have, with their discovery, the antibody engineering field is completely revolutionized. Okay, so this is how it looks like. So this is conventional antibody. So you have this different domains. So variable region heavy chain, right? Variable region light chain, which actually form the paratope. Here you have the paratope, which only is constituted by this VHH. Okay, then you have the hinge region and CH2, CH3. So you don't have CH1 domain here, so you have CH1 domain here, right? So this makes things light, rather very easy, in the sense that you can actually amplify only this segment and produce it in what we call as phase display libraries or lentiviral vectors, or and then you can modify a function of your proteins intracellularly, extracellularly. You can make it in different formats using bispecific or bivalent or multispecific, multivalent. So all those possibilities exist. Okay, so you can even make these antibodies in such a way that basically they bind to antigen on one side, okay, and on the C terminus you can actually put a toxin. So for example, if you have lymphoma actually is a cancer of B cells of particular type. So if you want to treat that lymphoma, so you select an antibody of VHH type against the surface antigen of this cancer cell, and on other side, so you put a toxin. So now this is going to bind to those cancer cells, and toxin is going to eliminate them, right? So this actually has provided a lot of advantage over conventional antibodies. Now, basically, this is the SDS page image, which actually was shown for this antibody. Okay, why I'm showing this is because we are actually working on these antibodies in the lab, right? So what essentially I'm trying to show here is if you purify these antibodies, right, the full length antibody, and you resolve the purified antibody either in presence of tumor captoethanol or in absence of it, so which is going to reduce the disulfide bond, right? So that means you are actually going to detect a single polypeptide chain, although I have not labeled it, but this is actually close to 45 kilodalton. Okay, this is actually 45 kilodalton. Now, if you do not reduce it, then this becomes close to 100 kilodalton. Okay. Unlike your conventional antibodies, where in reducing conditions, you are going to get heavy chain and light chain, and in unreducing conditions, you are going to get 150 kilodalton band. Okay. Just to basically tell you that these unique kind of antibodies also exist naturally, and they actually have revolutionized the whole field of antibody engineering. And why it was important to introduce these antibodies is because if you actually read any immunology book, which actually are recent versions, you will actually find the mention of these antibodies, which actually are naturally occurring heavy chain only antibodies. Okay. All right. So the next topic we are going to talk about is how on the earth you are able to generate antibodies which recognize every single antigen available in this universe. Okay, so you don't have so many genes to encode for it, right? But still, with this recombination event that is occurring both for actually BCR, B cell receptor, and T cell receptor, so you can generate antibodies or T cell receptor which can recognize almost unheard number of antigen specificities. This process actually is called generation of antigen receptor diversity. Okay, we are going to actually discuss this. If you want, maybe we can postpone it until the next week, or we can continue with this topic. And why I'm saying this thing is because quite a few of you are actually not present. So now tell me, so what do we do? So we continue with it? And if you don't continue with this topic, so maybe we can take up your questions and try to resolve your doubts in next 5 to 10 minutes.
Recording stopped.
Sir, could you please revisit on the functions of aminopterin?
Yeah, okay, fine. Yeah, so basically aminopterin, as I said, is a chemical basically. It's a antagonist of folic acid, and this folic acid is very critical for generation of nucleotides through de novo synthetic pathway, as was shown here. Okay, so you have this carbohydrate, basically conversion of the carbohydrate CHO from tetrahydrofolate. So this process actually is blocked by this antifolate, or in other words, aminopterin. So these intermediary steps are required for the conversion of PRPP into the precursors of nucleotides, inosinate, right, or inosine monophosphate. Now, aminopterin is basically acting here at this step and at this step. Okay. Now, if these steps are not present, I really did not talk about all these events which are actually occurring. So you can actually talk about, or you can actually go to any textbook for the nucleic acid synthesis, purine and pyrimidine metabolism. So you'll find all those steps, right? What my purpose here was to demonstrate to you that with the inclusion of aminopterin, so de novo synthesis pathway is blocked, right? And there is another pathway which, even if this pathway is blocked, can be utilized for synthesis of these nucleotides, right? This is called cell-based pathway. However, for this pathway to be utilized, so you require the activity of this enzyme HGPRT. Okay. Now, HGPRT should be absent, so its function should be missing in your cancerous cells, which you are using for hybridoma technology. Yes?
Yes, sir.
Yeah, any other thing? Actually, you can read through this slide if you want. So I'll give you, let's say, a couple of minutes time. Just read it. Okay, so clinically relevant pathway for synthesis of pyrimidine and purines. And this property is utilized for selection of your hybridoma. And that is why you have to actually select your cancer cells which are deficient in the enzyme which can utilize salvage pathway. And obviously, you are including this chemical, aminopterin, in the medium for selection, so which allow to block de novo synthesis pathway. So that means you don't select only the cancer cells, because cancer cells can always grow more than even your hybridoma cells, right? So even if you have contamination of one cell out of 100, so after a few rounds of division, so whatever you are left with is all cancer cells, and they are not going to produce antibodies. So you have to actually make sure that you select only the hybrid or the fused cell of B cells and cancer cells. So you have to actually get rid of only the cancer cells. You have utilized one property, which is actually to provide immortalization to your hybridoma cells, right? From that is coming from cancer cells. Apart from that, you don't require anything else from these cells. But actually, this was a revolutionary technology when it was discovered. Okay, um, uh, because it allowed scientists to select the specific antibodies, and you can make almost inexhaustible supply of these antibodies if they are clinically relevant to treat one kind of cancer, other kind of cancer, to neutralize infectious agents, to serve as diagnostics, to serve as biological agents or reagents in different analyses. So this whole thing actually came from the discovery of hybridoma technology. Very important, right? Okay, so more questions? If not, so what do you want me to do? So should we stop here, or I think let's stop here and we'll continue on this diversity part in next week. Essentially, these are the questions we are going to address. How does antibody diversity arise? What causes difference in amino acid sequences in the variable region? And how can different heavy chain constant region be associated with the same variable region? Okay, so these are all the questions we are going to address. But let's not complicate things today, so because it may not be so easy to understand. And I think we should continue with this topic when all of you are present. Okay, so I'm going to actually stop this session now.
Excuse me, sir.
Yeah.
Sir, when we were talking about the heavy chain antibody, you told about the VHH variable region.
Yeah.
So, sir, is there any is there any difference between the sensitivity of the variable region of heavy chain antibody in comparison to our conventional antibody?
What would you think?
Sir, I feel that conventional antibody will have much more sensitivity.
And you are actually right. Okay. any logical thinking person would like to think it that way. Because in conventional antibodies, so you have the paratop, which is consisting of 3 CDRs coming from light chain and 3 CDRs coming from heavy chain. So now you actually have these 6 CDRs which constitute the paratop, which can bind to antigen, right? So if your light chain is missing, so you are only left with this. So that means the combinatorial diversity that you get as a result of this combining with the variable region of the light chain and the heavy chain, so that is lost. However, in the VHH part, okay, one of the CDRs, rather 2 of the CDRs, CDR2 and CDR3, are quite extended, right? If they are quite extended, so that means they can actually compensate for the lost combinatorial diversity that is provided by the light chain. That's one. Okay. Second thing is, now in this VHH, okay, so normally the paratop is like this. This is kind of, okay, so this actually a form of pocket where your antigen can actually sit very nicely. Okay. However, with the crystal structure determination, it was shown for VHH that in some of the clones, actually, you can actually make a very projecting paratop. Okay. So you have this paratop, but because of the extension of the amino acid in CDR2 and 3, so you actually get an extension. Okay. So this actually can serve as a projecting or a pointing paratop. And why this is required? For example, for blocking the activity of many enzymes, and enzymatic active site actually are present very sequestered. Okay, they are actually hidden. Okay, and they are not really accessible by the conventional antibodies. If you want to block the activity of an enzyme, so probably this projecting paratop actually is much more required, so that this can actually pierce, this can actually bind those hidden epitopes as well. And in so doing, it can actually block the enzymatic activity. Okay, and obviously, so you have other advantages, which are basically you get excellent yield when you produce these antibodies recombinantly, even in bacteria. You can actually make a refolded antibody produced in E. coli. Forget about doing this thing for the conventional SCFVs in bacteria. So for that, you have to use yeast or eukaryotic expression system, and eukaryotic expression system has its own problem. You cannot really, I said inexhaustible supplies, okay, but that actually is a hyperbolic statement. Okay. There is always limitation in how much of antibody you can produce, but with bacteria, so you can produce whatever the amount you want, and whatever the antibody you produce. So if you actually choose right conditions, so this can be fully folded, and you don't have to do all these downstream events. Just purify your antibody and start using it. Okay, so several advantages have been associated with only having the single domain. Well, maybe I can point out, actually we got a center for antibody engineering from Department of Science and Technology, precisely on these single domain antibodies. Okay, so we are going to make antiviral, anti-cancer, different kind of antibodies.
Right, sir. Thank you.
Any other question? Okay, if not, so I'm going to upload these slides until monoclonal antibodies. I'm going to put on the Moodle page if I'm able to do it today, but obviously by the end of this week. Go through these slides if you have any questions, so we can discuss on Monday, right? And from Monday, so we are going to start the next topic, the diversity of antibodies, and obviously TCR. Now I'm ending this session.